Hello, everyone. Can everyone hear me? All good? Okay, cool. Hi, everyone. My name is Mark Kazour. I'm in the Sietz lab, Lindsay Seitz's lab at Northwestern University, and I'm a third-year graduate student. And today, I'll be discussing enhanced calibration techniques and design considerations for DEMS. So as we all know, the urgency to decarbonize our energy systems has pushed electrochemical transformations into the spotlight, where water electrolysis, CO2 reduction, hydrogen evolution, and nitrogen fixation could offer sustainable routes to fuel and chemical production. With recent adoption of renewable energy sources, energy has become cheap and abundant. Harnessing this new renewable energy, electrolyzers can be utilized to perform electrochemical transformations necessary for a wide range of commodity chemicals. To assess the production and distribution of these transformations from electrolysis, more traditional methods such as gas chromatography or nuclear magnetic resonance require significant concentrations for accurate measurement. Additionally, these measurements remain ex situ, and as these dynamic activity and selectivities of these catalysts will be lost, which also fluctuate with even constant current or potential runs. Further understanding of product distributions throughout operation is needed to inform catalyst material design, reactor operation, and reactor environmental control, leading to improved activity and selectivity towards a wide range of critical fuels and chemicals. So differential electrochemical mass spectrometry, which we've heard about a lot today, is an analytical in situ technique that captures product distributions at the site of production in real time. DEMS combines an electrochemical reactor with a mass spectrometer where volatile, gaseous, and liquid reactants and products are collected and quantified by continuous vacuum drawn through a pervaporation membrane. DEMS identifies these volatile species through a mass-to-charge ratio that is measured by a mass spectrometer. DEMS is a useful tool in measuring volatile species at a catalyst surface placed near or directly on top of a pervaporation membrane. And by operating at very low residence times, gaseous products can be quantified by measuring the required mass-to-ion currents. The relative amounts of products can be compared using the mass-to-charge ratio signal, and comparison between parallel DEMS experiments is possible if, and only if, the given parameters are consistent and reliable. But converting the intensity of DEMS signals to concentrations that can be compared to other methods remains a challenge. This is partially due to the normalization of vacuum pressure impacts the partial pressures measured, making it not possible to compare between different techniques. So such product calibration is necessary to form accurate kinetic information. To ensure consistency and proper quantification, cell design must be optimized, and reporting is very necessary. In a single chamber cell, like the one shown here, the working, counter, and reference electrodes are all inserted into the single chamber as shown. Many commercial cells, such as the Hiden A cell, demonstrate this design. A working electrode, most commonly a glassy carbon rod, is inserted into the top of the cell, and the electrode is positioned carefully so it is parallel and close to the evaporation membrane. Between the working and the membrane, a thin stream of electrolyte flows where the reaction of study occurs. Ports for the counter electrode and the reference electrode exist on small capillary junctions. Flow is optional with ports along the top of the chamber. The cell is very simplistic in design and compatible with many ubiquitous electrode designs. However, there lacks standardization in the working electrode distance from the evaporation membrane, which could result in signal sensitivity variation and difficulties in quantification due to differences in electrode placement. This cell design also limits the working electrode geometry and does not allow for separation of the working and counter electrodes into different chambers. And thus, products produced at either electrode may be measured, with products formed at one electrode can also migrate to and react at an opposing electrode. In a membrane-separated cell, we avoid product crossover and ensure consistency in the electrolyte path. In these cells, electrolyte often enters the working electrode chamber through a small inlet and then passes in a carefully controlled flow pattern over a working electrode. After reacting at a porous working electrode substrate, products pass through the evaporation membrane and enter the DEMS. The working electrode chamber is separated by an ion-conducting membrane to minimize product crossover, and this design also supports parallel counter and working electrodes. This design ensures consistent distance between the working electrode and the evaporation membrane, controlled electrolyte flow, as well as product separation. However, the highly specific working electrode geometry employed by many pre-configured cell options limit the choice of working electrode material. Our group develops more versatile flow cells by depositing catalysts directly onto the evaporation membrane or onto porous electrode substrates such as carbon paper or titanium felt. DEMS is employed across a variety of fields, including batteries, corrosion, and electrocatalysts, to gain an understanding of electrochemical phenomena. Today, I will focus on the use of DEMS for electrocatalysis. And in this talk, I will discuss a rigorous quantification method called the staircase calibration method, using the dual chamber cell under varying cell, catalyst, and substrate designs, as well as operating controls. Our aim is to standardize the quantification process and showcase the implications of multiple parameters have on cell performance and DEMS signals. This is very similar to what Ezra talked about today, but hopefully I'll enlighten you in a different way. So first, let's discuss a little bit more of this quantification method. A ubiquitous metric applied to electrochemical reactions forming multiple products is Faradaic efficiency, which conveys the fraction of total Faradaic current that is directed towards formation of individual products. For the CO2 reduction reaction, we employ a silver catalyst that is expected to produce only two products, carbon monoxide and hydrogen gas. As shown here, when applying a reductive potential, we drive carbon monoxide production and hydrogen production shown in these bottom figures. CO2 signal shows depletion from the baseline signal, indicating we're doing CO2 reduction. Using DEMS, the Faradaic efficiency of species can be indirectly calculated if all the generated species are known, such as those in the staircase calibration method. So the staircase calibration method is performed by isolating a single reaction, like the hydrogen evolution reaction from CO2 reduction reaction, and performing a series of CPs at more negative currents while measuring the mass over charge ratio of the products via DEMS. As we apply more reductive current steps, we can see a hydrogen DEMS signal response from a baseline product signal shown here. Taking the current response, we can develop a calibration slope for the hydrogen signal by plotting hydrogen DEMS signal versus current. And each current step, we take the last 30-second holds and average the response in the forward and reverse directions. And at each potential, we can develop a calibration slope. So once we've done this, the reactant for a second reaction can be introduced by bubbling gas or adding a soluble component into the electrolyte, which introduces carbon monoxide as a product. So taking the hydrogen calibration slope, we can subtract the current response from the total HER current response when 100% Faradaic efficiency is going towards hydrogen evolution to get the Faradaic efficiency towards HER in the CO2 reduction case here. And the rest of the current can then be attributed to CO2 reduction. And we can develop a Faradaic efficiency towards CO varying with current, like shown in this graph. And although it is important to note that because CO2 can fragment upon analysis by the mass spectrometer, it can fragment to CO, causing the signal of 28 for carbon dioxide. And like Ezra discussed earlier, we are able to calculate a known percentage rate that this happens. And in this case, it's 9%. And so the true amount of carbon monoxide created from CO2 reduction reaction is actually just 91% of the blue signal shown here. So another way to look at this is through oxidation reactions. So the same staircase method can be applied to oxidative reactions like the chlorine evolution reaction. However, unlike carbon dioxide reduction reaction, in aqueous systems, chlorine evolution competes with oxygen evolution, especially at higher current densities where oxygen mass transport limitations inhibit the transport of chlorine anions to the electrode surface. Therefore, quantification of CER products relies on first quantifying the OER without the presence of chlorine anions, as shown here in gray. Chlorine can then be directly measured with a mass to charge peak of 70. So first, starting with the absence of chlorine anions, we see a really nice staircase for the OER and no chlorine signal. The measured Faradaic efficiency of all current cases indicates 10% or less towards CER as shown in the Faradaic efficiency graph. And this small deviation is likely caused by residual chloride within the system and indicating that our method is relatively robust. So once we increase our electrolyte concentrations, we notice that OER signal declines to 0 at 0.5 molar case, shown in orange, and our chlorine signal increases. However, unlike CO2 reduction, chlorine products are complicated by the presence of multiple equilibriums between chlorine gas, hypochlorous acid, and the chloride anion. These equilibrium externalities cause the chlorine evolution curve to become much more complicated, as shown in our chlorine DEM signal that lacks the clear staircase that we see at for our oxygen evolution signal at 0 molar. And this highlights that trying to utilize our chlorine curves to quantify Faradaic efficiencies may not be an effective strategy. On the other hand, our oxygen results in the more ideal stepwise DEMS signal can more accurately average at each step in our 0 molar case. So by utilizing the OER staircase and developing a calibration curve when no chlorine anions are present, we can obtain a partial current response from the chlorine evolution reaction, and the rest of the current can be attributed to chlorine evolution. So when plotting the chlorine Faradaic efficiency, we can see that with increased chlorine concentrations, which clearly shows electrolyte has a significant impact on Faradaic efficiencies, with 0.5 molar sodium chloride having 100% Faradaic efficiencies across all current ranges. But at 0.1 molar, we see this deviation from 100%. So this data showcases that our quantification method requires standardization, and what other variables are we missing that could potentially impact our quantification strategy? So next, I'm going to enter the second half of my talk, where I'll discuss the parameters that greatly affect our calibration process. The variables I will discuss include electrolyte flow rate, chamber flow pattern, catalyst and substrate design, catholyte reservoir volume, and electrolyte concentration. These parameters greatly impact cell sealing, bubble formation on the electrode, and consistency of local reaction conditions. In order for us to quantify and achieve a replicable calibration, discovering the optimal conditions and reporting these design parameters is necessary. So first, I will discuss the impact of electrolyte flow rate has on Faradaic efficiency towards CO production for the CO2 reduction reaction. So when comparing the flow rates of electrolyte, we see a positive correlation of electrolyte flow rate and CO Faradaic efficiency. At lower flow rates, such as 10 skim, shown in gray, the lack of agitation leads to more bubble buildup at a catalyst surface, leading to lower accessibility of active sites directly on that catalyst surface. And so this is seen in our CPs with lower activity amongst the lower flow rates. Also, with slower flow rates, there is a decrease in mass transport of carbon dioxide to the catalyst surface. And ultimately, higher flow rates are ideal for improving reactant availability and limiting bubble development on our surface. So we also expect this trend to remain relatively valid for many reactions involving gaseous products. So as we've seen, electrolyte flow rate can dramatically impact our Faradaic efficiencies. So now we'll discuss more on electrolyte concentrations and their impact on the quantification process. So when comparing the electrolyte of potassium bicarbonate in our carbon dioxide reduction reaction system, we notice higher carbon monoxide Faradaic efficiencies in all cases with lower concentrations of potassium bicarbonate. For comparison, the percentage of carbon dioxide availability versus the ideal case, which is 0.1 molar potassium bicarbonate, we see only a 34% availability of CO2. And we basically just take the baseline curve for CO2 in an ideal case versus the not ideal case and compare them. And that's how we get our ratio here. And this is a result of the solubility limits of CO2 under high concentrated electrolyte systems. And therefore, we really have to consider solubility limits when optimizing the entire system. So in our chlorine evolution case, we show a strong dependence of on sodium chloride concentration. Interestingly, all runs with sodium chloride yield 100% Faradaic efficiency to chlorine at low current densities, and with low concentrations of sodium chloride correlating to decreasing Faradaic efficiencies upon higher currents. At the maximum current tested, 5 milliamps, the 0.1 molar sodium chloride shows a Faradaic efficiency of only 70% towards CER, while 0.5 molar sodium chloride maintains that 100% Faradaic efficiency. And this suggests that the system becomes mass transport limited at sodium chloride concentrations of 0.25 molar or lower, guiding us to select the most ideal electrolyte to maximize CER. So as we've seen, electrolyte concentration can vary the Faradaic efficiencies of our desired products, and that electrolyte concentration can vary depending on our system of investigation. So now we'll discuss the reservoir volume and how it impacts our quantification. So when considering reservoir volume for the CO2 reduction reaction case, we analyzed only varying the volume of the reservoir and the residence time in the reactor where the EChem and DEMS detection was performed remained constant. So we see that 1 liter results in higher carbon monoxide Faradaic efficiency in almost all flow rate cases, except for the 10 skim case with a smaller reservoir volume. The electrolyte is not fully saturated with CO2 as it is returned to the cell, meaning CO2 saturation is not optimized at lower reservoir volumes. With a large reservoir, the electrolyte has a longer residence time and the dilution rate of CO2 is lower. So in the 10 skim case, the electrolyte has a longer residence time to counteract the size of the reservoir. A larger reservoir may lead to dilution of CO2 at such low flow rates, which cause the overall decline of carbon monoxide Faradaic efficiency. Therefore, reservoir volume impacts the saturation of CO2 within the electrolyte and transport of CO2 to the catalyst surface. Chlorine evolution, we didn't perform this because we weren't involving a gaseous reactant. And so as we've seen, system considerations can impact Faradaic efficiency and quantification measurement. However, proper cell design also can have a significant impact on quantification. So next, I'll discuss electrolyte flow pattern within the dual chamber DEMS cell. So to study the effects of flow pattern on CO2 reduction, 2 cell designs were developed. The first one is a more laminar design with consistent flow fields drawn into and out of the main chamber. The second is a more optimized case with an impinging jet flow path that supports turbulent flow within the reaction volume. In the turbulent design case, shown at the bottom here, okay, yeah, can't see my cursor, but the pathway points down towards the catalyst surface, which aids in replenishing reactant directly at that catalyst surface. And so our results indicate that turbulent flow, all electrolyte flow rates and amongst all currents improved carbon monoxide Faradaic efficiency. This is a direct result of CO2 constantly being replenished while eliminating bubbles on our catalyst surface. The laminar flow design, on the other hand, provides less mixing of the flow pattern, which limits concentrated carbon dioxide electrolyte from reaching the catalyst surface, and it also doesn't remove bubbles. And so the measured voltage generated here also indicates a higher overpotential for the laminar design, which further supports our conclusions that faster kinetics happen under our turbulent system. And therefore, flow fields are critical in supporting optimal mixing, transport, bubble removal, and faster kinetics. And lastly, not only will flow patterns impact Faradaic efficiencies, but also catalysts and substrate design. So here, we compared the impact of catalyst deposition directly onto a membrane versus on an adjacent porous conductive substrate, such as carbon paper for carbon dioxide reduction. Essentially, we aimed to see if utilizing different electrode morphologies that more closely resemble those employed in more applied devices, such as a gas diffusion electrode, could be applied to our cell design. In the carbon paper case, a silver catalyst is facing up, as shown in this figure. And as shown by our carbon monoxide Faradaic efficiencies, deposition directly onto the PTFE membrane yielded a higher Faradaic efficiency for CO than the carbon paper case. So these figures depict that the more reductive potential with reductive current indicates mass transport limitations with the carbon paper case. Even though it is possible that higher carbon monoxide Faradaic efficiencies is occurring on our catalyst surface for the carbon paper, it has to travel through the carbon paper and the PTFE membrane to reach the DEMS detector. Reactant and product can become trapped within the carbon paper and leading to our lower DEMS signal. So it's important to note that some of the calculated Faradaic efficiencies towards CO are negative. And this also indicates that our hydrogen calibration curve method was not consistent in our carbon paper case as the PTFE deposition case. And this is due to the lower mass transport limitations when just hydrogen is being produced versus when both hydrogen and carbon monoxide are being produced simultaneously. So overall, the direct deposition on the PTFE membrane is the more optimal case here. And so lastly, I would love to conclude that DEMS is an invaluable tool in quantifying reaction Faradaic efficiencies with techniques such as the staircase method and revealing important reaction dynamics such as mass transport limitations in real time. We demonstrate that the quantification of DEMS for CO2 reduction, first through the analysis of a simplified hydrogen evolution reaction staircase, as well as for chlorine evolution by first taking the simplified oxygen evolution staircase. And they both isolate individual products, and then gradually we build up in complexity. So we highlight the importance of examining numerous system conditions, including electrolyte flow rate, electrolyte concentration, catalyst and substrate design, and reservoir volume. Finally, we underscore the importance of cell parameters like scan rate and hydrodynamic flow fields in cell design. And with these considerations, novel system designs can be realized that maximize reactant transport and product collection efficiencies for a suite of chemical reactions. The cell design conditions and quantification techniques are necessary to broaden the applicability of DEMS to other electrochemical systems and enhance reproducibility among literature. Although the staircase method provides a useful benchmark for quantifying product distributions in real time, further calibration strategies for quantifying complex reaction environments with numerous redox reactions, equilibrium conditions, and non-volatile products are necessary. With enhanced spectrometric calibration techniques and design considerations, electrochemical mechanism schemes can be identified and inform future catalyst design for industrially relevant scale-up. As electrochemical methods and techniques such as DEMS are refined, sustainable electrochemical solutions that bolster climate-conscious design can be realized. And so lastly, I would love to give acknowledgements to Professor Lindsay Sietz, my PI, for the tremendous mentoring support throughout this project, as well as Kavi Chittum, AJ DeBurgess, and Jane Edgington, without whom this project couldn't have been realized. And I'd like to thank my funding sources, and I'm happy to take any of your questions.
Thank you, Mark. Do you have any questions from the floor?
Thanks for the nice talk. I got a couple questions for you.
Yeah, sure.
For first one, have you thought about actually quantifying the hydrodynamic boundary layer thickness using your impinging, comparing the impinging jet morphology to the kind of standard just straight tube morphology using something like ferrocyanide reduction?
Oh, yeah. No, I think the direction we've been going with this project is trying to quantify how turbulent the flow is, and trying to partner with some groups that have more experience in modeling this. But I would like to pick your brain more on that. We haven't really considered that.
Sure. And then my other question was about the carbon paper result. You showed the CO Faradaic efficiencies. How does the total Faradaic efficiency compare with the carbon paper? And do you see any sort of bubble noise in the mass ion currents when you're using the carbon
noise when using? Oh, yes, definitely to the bubble noise. And I think purging with our electrolyte, especially with our pump in the positive direction of flow, causes a lot of those bubbles. And so we have been trying to adjust our flow rates to counter that. And then as well as the total Faradaic current, we do also produce carbon dioxide throughout the process. And so that's another constraint for this particular substrate. And we've switched to other substrates like titanium felt to try to counteract some of those unwanted reactions that are happening at our surface. So, yeah.
My question was sort of similar. You call the. I'm a little confused. I need a little bit of clarification.
Yeah.
In your turbulent flow reactor, the only thing that's different is these angled ports now?
Yes, that's the only change.
So how are you classifying that as turbulent flow, though? Like, was there any modeling that was done that's now showing that this is actually
turbulent flow?
Yeah,
yes.
Sure.
We did calculate. As someone that has come from Penn State University, I do know how to calculate a Reynolds number. And we did do that for these cases. And yeah, that further supported that we were discussing turbulent flow for our turbulent case.
I mean, one thing that you could do, as a Penn State grad is then go and calculate the Sherwood number and use flow over a flat plate and get a convective mass transfer code.
Yeah, no, for sure. Love the suggestion.
Yeah.
And yeah, we'll consider that for sure.
Cool. Yeah, we've seen pretty good agreement between what you measure with ferrocyanide reduction and just assuming flow over a flat plate and using a Sherwood correlation, which is why I asked.
Okay, yeah.
Yeah,
thank you, yeah.
For your the silver titanium that you have?
So silver, in this case, we electron beam deposition silver onto a PTFE membrane for this study.
You have this titanium adhesion layer? Is that an adhesion layer?
Yeah, so we have just carbon and then this small tie layer. I believe this is a mistake on this figure. I don't believe we have this small tie layer. It's just bare carbon separating the electrolyte as well as the PTFE membrane. Yeah. And then we did do studies with just bare tie felt as well as cobalt oxide on tie felt. So yeah, we've kind of been exploring our substrates a little more.
Okay. I thought it
Yeah.
was titanium. That's why I was asking.
Oh,
But it's not titanium. Okay.
okay.
Thank you. Thank you.
There is a question from the chat.
Sure.
Why is the concentration of KHCO3 affecting the CO Faraday efficiency?
Yeah. This slide. Yeah, this slide. So there is like this relative CO2 dissolution that happens when your concentrated system is too concentrated. It leads that CO2 to be leached out of your electrolyte, and then you're no longer having CO2 being replenished at the electrode surface at a higher rate than if your concentration of potassium bicarbonate was lower. And so you have to consider how much CO2 is actually being able to be taken up by that electrolyte. And so this case just highlights that solubility limits are really important when optimizing our system.
We got a new one that just came in here. Can you confirm that the use of carbon paper only affects the data quality due to the path the analyte must follow through it, and not a technical issue regarding electrolyte flooding into the mass spec, like Ezra previously said?
Yeah, so I guess flooding into the mass spec from the electrolyte, is that what you're trying to ask, or? Yeah. So the prevaporation membrane is preventing the electrolyte from flooding the DEMS. So we don't have the carbon paper is resting atop the prevaporation membrane.
Okay, all set? Mark, thanks so much again. Thank you.